Okay, so moving on to our final section, section 11, where we're going to talk about how each one of our macromolecules is actually processed. When we're talking about digestion, we're talking about the breakdown of these macromolecules, proteins, carbohydrates, lipids, and nucleic acids, into their monomers. And then how do we get these monomers that are in our lumen of our small intestines across the enterocyte and into our blood? That's the absorption part. When we're talking about digestion, just as kind of a refresher, most of this is going to occur in our small intestine. We have pancreatic enzymes that are helping with the breakdown, but then the final breakdown processes are actually occurring via those brush border enzymes in the intestinal wall. We also have pancreatic juice, which is alkaline, so meaning slightly basic, and it's going to help neutralize that acidic chyme that's coming in from the stomach. We also have lipases and bile that are going to help with fat breakdown. When we're talking about digestion, again, taking a macromolecule and breaking it down into its chemical building blocks or monomers, that process is considered a catabolic process. So catabolic is breaking things down, anabolic is building things up. This catabolic process of digestion is also called hydrolysis because it's going to require water. So for absorption as an overview, this process is involving getting the substances that we're breaking down that are in our intestinal lumen across the enterocyte, or the cell that's making up the wall of the small intestine, into the interstitial fluid, and eventually into our bloodstream, where we can transport those nutrients to various parts of our body. In between each one of our enterocytes, we have tight junctions. And the purpose of these tight junctions is so that we do not have spread of ions or different things between the adjacent cells. The only place we want substances to be able to cross the plasma membrane of an enterocyte is on the apical surface and the basolateral surface. So the apical surface, we're talking about the brush border, the microvilli. This is the side that's facing the lumen of the small intestine. Our basolateral surface is the one that's in contact with the interstitial fluid. So we're going to be moving substances across the apical surface, going through the enterocyte itself, and then out of the basolateral surface into the interstitial fluid. If we're talking about our macromolecules, our lipids are lipid soluble, which means that they can directly cross the plasma membrane of the enterocyte. Most of them can just diffuse directly across. If we're talking about our other monomers, most of those are going to be polar substances, which means they cannot directly diffuse across the plasma membrane. They're going to need some sort of transport protein to help them cross, and most of the time this transport protein is going to require ATP. So they're going to be absorbed by active transport. Again, when we're talking about absorption that's primarily occurring in the small intestines, it's not occurring along all three of those subdivisions of the small intestines. Most of the absorption is only occurring in the duodenum and the jejunum. When we're talking about the ileum's role, its role is really in reclaiming the bile salts. So remember, we talked about recycling the bile salts back to our liver. Well, most of this is going to occur at the end of the small intestines at the ileum. So let's start with our first macromolecule, which is carbohydrates. Most of the average diet is carbohydrates, and most of the carbohydrates that we're taking in are in the form of starch. However, even though these are the carbohydrates that are coming in from our food, we can only absorb across our enterocyte monosaccharides. So we have to be able to break these carbohydrates down into individual carbohydrate monomers, which are called monosaccharides. Sometimes we do have monomers that are linked together. If we have monosaccharides that are linked together, these are just called disaccharides for two, mono for one. If we're talking about monosaccharides, we have three common ones in our diet. Those are going to be glucose, fructose, and galactose. The common disaccharides from our diet are sucrose, which is really just table sugar, lactose, we're talking about the sugar from milk, and maltose, which is a grain sugar. Our polysaccharides are glycogen, which is the animal storage of carbohydrates, and then starch, which is the plant form of carbohydrate storage. So this is a nice overview slide where we're talking about digestion of carbohydrates. Remember, it's going to start in our mouth because we have salivary amylase. However, this isn't going to accomplish all carbohydrate digestion. We're only really contributing little or minor breakdown of our carbohydrates. It's not really until we reach our small intestine and we have access to the pancreatic amylase that we are going to be doing the bulk of the carbohydrate digestion. So two sources or two locations in which we do break down carbohydrates, starting in our mouth, and then the second place is the small intestine. However, both the salivary amylase and the pancreatic amylase really only break down these polysaccharides into oligosaccharides and disaccharides. Oligo just means a few or a couple of monosaccharides linked together. So still pretty small, they're just small fragments of linked monosaccharides, but we still don't have our complete single unit monomer broken down yet. It's not until we actually hit those brush border enzymes that are in the microvilli of our small intestine where we're going to take oligosaccharides and disaccharides and actually be able to break them down into individual monomers or monosaccharides. So this is just kind of like a reference slide. We've already talked about this before. We have our pancreatic amylase that's going to break down our polymers, so starch and glycogen. Any of the polymers of carbohydrates that have escaped the salivary amylase will be broken down once they hit the small intestines. And then the brush border enzymes are going to accomplish the breakdown of those oligosaccharides and disaccharides into the individual monosaccharides. So the only thing that can actually cross the enterocyte are monosaccharides. So we have a monosaccharide right here. An individual hexagon in this image over here represents a monosaccharide. Over here you can see we have two hexagons linked together, so that represents a disaccharide. Hasn't touched the brush border enzymes yet, so it's still linked together. And then once it reaches the brush border enzymes, we can cleave the bond between them and get our individual monosaccharides. In order to get the monosaccharide from the intestinal lumen and into the enterocyte, we actually need another important ion. So here's our transport protein. If you notice at the image, we also have sodium that's entering in this transport protein along with our monosaccharide. So in order to get a monosaccharide into the enterocyte, it actually has to be co-transported with sodium. This co-transport is going to be a form of active transport, meaning it does require energy, but energy indirectly because it is secondary active transport. Once it's inside the enterocyte, it's going to exit the basolateral surface at this protein, transport protein right here. It doesn't need to be co-transported with anything. It's just going to simply diffuse across the plasma membrane through this channel. So that's going to be facilitated diffusion. Eventually, the monosaccharides will enter the interstitial fluid and then into our bloodstream. Our next macromolecule are proteins. The monomers, or the building blocks of proteins, are amino acids. So we do break down our proteins into what we call large polypeptides, where we still have several amino acids linked together, and then eventually some small polypeptides, and then small peptides, and finally into the monomers that we want. If you remember back to our digestive system, protein digestion begins in the stomach once we're able to get the active form of our enzyme required to break them down. So once we convert pepsinogen into pepsin via the hydrochloric acid, then we can go in and start protein digestion. Here's our little recap table. We have our active form of the enzyme, so we have pepsin right here. So we begin protein digestion in our stomach. We then continue on with our food product that's going to enter into our small intestine, and then we have pancreatic enzymes that are going to come into play to help further break down those large polypeptides into smaller peptides and polypeptides. It's not until we actually reach the brush border enzymes of the small intestine that we're able to break down those small peptides and polypeptides into individual amino acid units. Again, this is kind of a recap of some of the processes and how the pancreatic proteases work. What we're concerned with in this slide is how do we get an amino acid from the intestinal lumen across the enterocyte and into our bloodstream. That is what absorption is. So if we look here at the apical surface, we see a protein in the plasma membrane. These individual gold balls here, these represent amino acids. Just like we saw with our monosaccharides, we actually have to co-transport amino acids along with sodium in order to get them into the enterocyte. Once they're in the enterocyte, they can exit via the basolateral side using a protein carrier. So this is just going to be a form of facilitated diffusion. And then eventually it's going to enter into the interstitial fluid and then our blood. So same thing, this is the same process. Protein and carbohydrates both co-transport into the enterocyte, facilitated diffusion out of the enterocyte at the basolateral surface. Okay, our next macromolecule is going to be lipids. Lipids are a little bit different. They have a slightly different process in order to get them into and out of the enterocyte. So we go through a process that's called emulsification. Because the triglycerides and their breakdown products are not soluble in water, they need what we call a pre-treatment. And this pre-treatment is basically incorporation of bile salts. So we also have our pancreatic lipases that are able to come in and break it down into its components. Remember, we don't have true monomers when we're talking about lipids, but in this case, we can break down triglycerides into what we call a monoglyceride and then two fatty acids. So monoglyceride is just a glycerol with one fatty acid chain attached, and then we have two free fatty acids. The next thing is that we form what's called a micelle. And a micelle is just really a collection of fatty elements that are clustered together with bile salts. And it kind of is organized in a way that the polar or the hydrophilic ends of the molecules face the water, and then the nonpolar portions form the core of it. So I'll show you a picture on the next slide. Then we have diffusion, where those lipid products actually leave the micelles and cross the epithelial membrane. Again, we just need diffusion because the plasma membrane is polar. And then we have chylomicron formation, where these lipid products are converted back to triglycerides and packaged together with other things to form a chylomicron. Then we have our chylomicron transport, where we exocytose these vesicles from the basolateral side, and then they're going to enter into the lymphatic lacteal and eventually empty into venous blood at the thoracic duct. Once they're in the blood, those chylomicrons are broken into free fatty acids and glycerol by an enzyme called lipoprotein lipase, so they can actually be utilized by cells. If we have just short chain fatty acids, those can just diffuse directly into the blood. So this is the steps. We have emulsification. That's our step 1 right here. That's where we have bile salts that are in the duodenum break up the large fat globules into smaller ones. So here's our large globule up here. Bile salts are going to come in and adhere to help break up this larger fat globule into these smaller ones. It's going to help increase the surface area for our lipases that we are going to encounter when we hit step 2 for digestion, when we're in the small intestine. The next step is micelle formation. These little green things are what are called micelles. Again, this is predominantly just fatty acids and some bile salts and some other things. This is what is actually going to enter in to the epithelial cell. So we're going to have fatty acids and the monoglycerides diffuse from the micelles into the epithelial cells. Once we have these in the epithelial cells, we're going to get chylomicron formation, where the fatty acids and the monoglycerides are going to be recombined and packaged with other fatty substances and proteins to form the chylomicron. The chylomicron is actually what is exocytosed from the basolateral side of the enterocyte itself and ultimately taken away into the lymph. Just a refresher slide. So we do begin true lipid breakdown, I guess a little bit of lipid breakdown when we're talking about in our mouth. We have a lingual lipase there. However, it's really, really minor, not really considered a huge amount. Once we hit the stomach, we also reach another lipase, so gastric lipase. So we do a little bit of lipid digestion in our stomach as well. However, just like with every other macromolecule, our small intestine is going to be the predominant location of breakdown of lipids. And that's accomplished by the bile salts and pancreatic lipases. So anytime you're taking in and digesting any sort of cell, we know that the nucleus of cells has DNA and RNA, so those are both forms of nucleic acid. We also have an enzyme called nuclease that can go in and break down nucleic acid. So this is going to occur when we're in our small intestines, and it's a pancreatic nuclease. It's going to break down either one of these nucleic acids into nucleotides, which are the monomers. We also have brush border enzymes that are going to help continue with the breakdown. So they can basically break down nucleotides into their three components, which is a pentose sugar, a phosphate, and a nitrogenous base. Then we're going to take those breakdown products and transport it across the enterocyte. So really, we didn't really mention nucleic acid breakdown in our previous chapters. We're focused on the other nucleic acids, other macromolecules that we have in our diet. But here you can see if we're talking about nucleic acid digestion, that is only occurring in our small intestine. And we're relying on those pancreatic nucleases in order to break these down, along with the brush border enzymes. Okay, vitamin absorption. The small intestine is absorbing the dietary vitamins that we're taking in. The large intestines absorb some of the vitamin K and B vitamins that are made by our gut microbiome. When we're talking about vitamins, our fat-soluble vitamins, which include A, D, E, and K, because they are fat-soluble, these have to be carried by those micelles, and then they're going to diffuse into absorptive cells. Our water-soluble vitamins, like B vitamins and C vitamins, are absorbed via specific active or passive transporters. Our one exception is B12. It's really, really big and it's charged. And remember, we need the intrinsic factor that's produced by our stomach to bind vitamin B12 and get it across our enterocyte. And we also talked about our large intestine's role in the vitamin K and B from bacterial metabolism being absorbed as well. Electrolyte absorption. So most ions are transported actively along the length of the small intestine. Actively just means requiring ATP or energy. Iron and calcium are going to be absorbed in the duodenum. You have to have vitamin D in order to absorb calcium. We also have sodium absorption. That's going to be coupled with the absorption of our monosaccharides and amino acids. We saw that previously when we were talking about protein monomers and carbohydrate monomers. Both of those had to be absorbed as co-transport with sodium. If we're talking about chlorine or chloride ion, that is going to always be transported actively, meaning that it requires energy. Typically, if we're talking about potassium, That's going to just require diffusion, and diffusion is a form of passive transport, which means that it doesn't require any energy. Iron and calcium absorption is related to how much of it we actually need as well. So we already talked about transport of iron when we're in the blood. So if we're talking about ionic iron, meaning that it's charged, this is stored in mucosal cells as ferritin. However, whenever we need to transport this iron ion in our blood, it's transported as transferrin, which is just a plasma protein that transports it and helps transport it in the circulation. Okay, water absorption. Approximately 9 liters of water, most of it is derived from the GI tract secretions, enter into our small intestines daily. Water is the most abundant substance in chyme, and 95% is absorbed in the small intestine by the process of osmosis, if you remember that from AMP1. Most of the rest of it is going to be absorbed in the large intestine. Net osmosis occurs if concentration gradient is established by active transport of solutes that we have, particularly sodium. And then water uptake is always going to be coupled with solute uptake. And we'll talk about this when we hit the urinary system and talk about the role of the kidney. So we can have something called malabsorption. This is just basically meaning that we have some sort of impaired nutrient absorption, and it has several different types of causes that are very varied, and it can result from anything that interferes with the delivery of bile or pancreatic juice to the small intestine. Other things can lead to it, like if we have damage to our intestinal mucosa, or if for some reason we have a reduction in the absorptive surface area. We have gluten sensitivity and enteropathy, also referred to as celiac disease, which typically affects about 1 in 100 people. This is a common malabsorption disease. So we have an immune system reaction to gluten, which is a protein that's found in grains. And the breakdown products of the gluten interact with molecules of our immune system and our GI tract, forming these complexes that are going to go on to activate T cells, which then attack the intestinal lining and damage our intestinal villi and things like that. So because of that, we get malnutrition. So treatment basically is to eliminate gluten from the diet.